stage, which we call as a preparatory stage, or the stage where the molecules are being prepared for the production of energy in the next stage, there are two ATP which has been utilized. So actually, if I look at this stage, there is no production of energy. It is just ATP is being consumed. And it's a very, different scenario where we call about that glycolysis is giving us energy. But in the first stage, energy is actually being consumed. And where are these two steps? First is where glucose is converted into glucose 6-phosphate, that is a hexokinase. And second one is where fructose 6-phosphate is converted into fructose 1,6-bisphosphate. And both the enzymes you see are kinase enzyme. One is a hexokinase and second is a fructose 6-phosphate kinase. So this stage we have already discussed. Second stage which we have discussed is where fructose 1,6-bisphosphate is cleaved into 3-carbon compound glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And these two, you know, they are interconvertible. They are converted from because both of them, they are isomer in nature. One GAP, glyceraldehyde 3-phosphate, is aldo group, and here you have a keto group. So both are 3-carbon compound. They are aldo keto isomers. And what happens because of the presence of enzyme isomerase, most of dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. And the conversion, both in terms of formation of dihydroxyacetone phosphate or GAP, or their interconversion, they are reversible process. And because this is reversible process, it is possible that they can form fructose 1,6-bisphosphate. As you know that the aldose can convert DHAP and GAP into fructose 1,6-bisphosphate. But because glyceraldehyde 3-phosphate is being utilized in the next few reaction, the reversal of the reaction doesn't occur. And most of this molecule, they are converted into next stage of glycolysis. So I have covered stage 1, I have covered stage 2. Today I'm going to talk about stage 3. And stage 3, in this stage, you will see that this is the stage where your ATP is being produced, number 1. Number 2, you will also get another molecule, which is NADH. What is the importance of NADH? NADH is the one which is having high electron donating potential. So NADH at later stage, when you will learn about oxidative phosphorylation, will be the one which will be donor of electrons. So ultimately this proton, which is extra proton, when they are released from NADH, the proton produced and the electron moves in the oxidative phosphorylation. What happens to proton? Proton, they are coming out of the mitochondria and they will generate proton motive force, and that will be the process by which your energy is going to be produced. That portion I will discuss separately. That's a different, discussion, how ATP is being generated. But here, what you find is that, now let us look at this. Stage 1, you have consumed 2 ATP. Stage 3, you have produced 2 ATP. But ultimately, what happens, because here at this stage, you have 2 molecules of 3 carbon, which is being produced from glucose. So the number of this part, stage 3, is occurring twice. And because it is occurring twice, so at the end of glycolysis, what you will get is that from 1 molecule of glucose, you will get 2 molecules of pyruvate, 4 molecules of ATP, and 2 molecules of NADH. And here you have utilized 2 ATP. So at the end, what is happening? You are getting 2 molecules of ATP. So this part, again, I will summarize and discuss. So let us go back and discuss the third stage of the metabolism. So this part we have covered. Now we are moving to the conversion of glyceraldehyde 3-phosphate. So now what you have learned is that glucose has been converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and they have isomerized to make 2 molecules of glyceraldehyde 3-phosphate. So one molecule of glucose is capable of producing two molecules of glyceraldehyde 3-phosphate. We have assumed that all molecule of dihydroxyacetone phosphate has been converted into glyceraldehyde 3-phosphate. Is this clear to you? Is this concept clear to you?
Yes, sir.
Yeah? Yes or no?
Yes, sir, it's clear.
Okay. So actually what I'm discussing is that suppose all the dihydroxyacetone phosphate is not converted into glyceraldehyde. In that case, what will happen? That the number will not be doubled. And why it happens? Why all the molecules they are converted into glyceraldehyde 3-phosphate? Though they are in equilibrium, but because it is being moving towards the forward direction, the reaction, all the dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate, as per you have learned in the Leach-Atelier's principle, that if I remove the product, the reaction moves in the forward direction. Okay. So what is this reaction? This reaction is happening in presence of dehydrogenase. And you will see that this aldehydic group is converted into first acidic group, and then a phosphoryl group gets attached. I will explain it. And second important thing is happening is that NAD plus, which is present here, is converted into NADH. So what is happening? What happens to glyceraldehyde 3-phosphate? This is the one which, when it is converted into acidic form, that means you are oxidizing this molecule. So oxidation of glyceraldehyde 3-phosphate occurs. What happens to NAD plus? This is getting reduced because it is forming NADH. So this step is very interesting step, and it happens in two stages. In the first stage, what happens that glyceraldehyde 3-phosphate reacts with NAD plus in presence of water, and oxidation reaction occurs. So what you get is you get 3-phosphoglyceric acid. So here aldehydic group has been converted into acidic group. That's the one conversion. And during this process, this NAD plus has been converted into NADH. So this water molecule, that is the one which is responsible for, this reaction. And let us see, I will explain the mechanism also. So first step, your NADH has been formed. Second step is where phosphorylation occurs. You have 3-phosphoglyceric acid, which reacts with inorganic phosphate, and you get 1,3-bis phosphoglycerate. So third position, one phosphate group has been attached. So end of the reaction, you have oxidation reaction, and you have production of NADH. So this whole reaction, if I look at, it is involving two things. One is oxidation, and second is phosphorylation. So we can call it that it is oxidative phosphorylation. Uh, what I'm trying to say here is, don't get confused when we talk about the next reaction, where oxidative phosphorylation is a coupled reaction. This is the one where a substrate molecule is the one which is source of energy for uh, you know, uh, uh, this is the place where you have a substrate which is converted in presence of Pi into a molecule which gets phosphorylated. And this phosphorylation, so you have generated a molecule which is a phosphorylated molecule. In future, these phosphate groups, they will be utilized for the formation of ATP molecule. And most important thing here is production of NADH, which in future is going to provide a high electron donating potential molecule. Let us see how actually happens. What is the mechanism? The mechanism, I don't want to take you to the greater details of the mechanism. This is just to show you that on the enzyme active site. So this is the site. What is the enzyme? Enzyme involved here is glyceraldehyde 3-phosphate dehydrogenase. So on the surface, you have a cysteine residue. It is at 149 position. This molecule, what is this molecule? This is the NAD plus. So on the surface, you have, and I have reduced the structure of glyceraldehyde 3-phosphate because R has been written, just only aldehydic group has been exposed. So here the reaction starts with the transfer of hydride ion to a molecule of, so first step is that your aldehydic group, whatever is present, it will join with the sulfhydryl group. And where is the sulfhydryl group? It is present on the cysteine residue of the enzyme. Now, once this bond is formed, then it starts step 2, where hydride transfer occurs. This is hydride ion will be transferred to NAD plus. And what will happen? This will become NADH. On the other hand, then rearrangement will occur, and then finally, in the next step, your orthophosphate, the phosphate group, that will attack, and you get 1,3-PPG. So this is the phosphorylation reaction. I'm not very much concerned about this. The major theme which I want to emphasize here is number 1, which I can explain by next slide. There are 2 steps involved. The step number 1 is oxidation process, and step number 2 is phosphorylation process. So this is the oxidation reaction, and this is a phosphate group formation. Now, if I consider that these two are separate reaction, there will be oxidation. There is a big difference in the free energy, and acetyl phosphate formation occurs, and at the end you get the enzyme products. But actually, what happens, because of the formation of thioester intermediate, those two reactions, they do not create a very big uphill for the reaction to happen, and both of the reaction, they are occurring on the surface of enzyme. As you can see here, that this is the surface of the enzyme, which first provides the binding of the phosphate group with the SH group, and then in the second step, there is a hydride transfer will occur, NADH. So once NADH is removed from the surface of the enzyme, then only the second step of phosphorylation occurs. So it is a step where you have oxidation reaction, and second step where phosphorylation. So this is the oxidation, and this is the phosphorylation. So look at this enzyme, glyceraldehyde 3-phosphate dehydrogenase. It is doing 2 jobs. Name is there, dehydrogenase. That means it is involved in oxidation process, but it also does the phosphorylation reaction. Okay. So now, after the phosphorylation, you have formed 1,3-bis-phosphoglycerate. So this molecule has been formed. So what was the purpose of forming this? The purpose of this was to generate high energy compound, which is ATP. And how it does? Because this has enough phosphoryl group transfer potential, which is higher than the ATP, and that's why its phosphoryl group can be transferred to ADP molecule in the presence of enzyme phosphoglycerate kinase. Again, you see that the enzyme is kinase because ATP and ADP is involved in the transfer from one form to another form. So what happens in this reaction? This is the almost final stage. There is one more step which will be required for the glycolysis. So here 1,3-bis phosphoglycerate, it will transfer its phosphoryl group, and then it becomes 3-phosphoglycerate. So you see, first in previous stage phosphorylation has occurred, here dephosphorylation occurred. Now in these 2 steps, what you find is that the difference is in the molecule, in this structure, which becomes, which has much more potential to transfer its phosphoryl group. But before that, suppose I take just 3-glycerophosphate, that will not transfer its phosphoryl group. So that's where it is important that this molecule should have been generated, and that is what glycolysis has done. So the outcome of the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase and phosphoglycerate kinase. So I'm taking the sum of these 2 reactions. First, previous step, and this step. Now glyceraldehyde 3-phosphate, which was an aldehyde, it was oxidized to 3-phosphoglycerate, and then NAD is being converted into NADH, and ATP is formed from ADP and PI. So ATP is formed in this step, and in the previous step, you have already seen that in this step there are NADH and formation of a high energy rich molecule. So now let me just give you some very basic idea that this is step where a substrate is involved in phosphorylation of ADP molecule is called as a substrate level phosphorylation. You will see one more reaction where substrate will be the one which will be transferring phosphoryl group in this glycolysis, and these two steps they will be concerned, they will be called as a substrate level phosphorylation. Other type of phosphorylation are called as oxidative phosphorylation. Do not get confused with the previous step which we have talked about, where oxidation phosphorylation occurs. This is the one where substrate is the one which is involved in the phosphorylation of ADP molecule. Now, you have got 3-phosphoglycerate. And as I said, that 3-phosphoglycerate molecule does not have high energy rich position where phosphoryl group can be transferred. And that is why there will be interconversion. And how the interconversion occurs? 3-phosphoglycerate forms 2-phosphoglycerate. And this is happening by an enzyme which is kind of isomerase enzyme. We do not call them isomerase because there are isomerase is a general name, and most of the isomerases are where a group is being changed. So you might have seen that aldehyde group transferring to keto group, that is called as isomerase. Here, wherever the isomerization reaction occurs by transferring one group from one position to other position is called as a mutase. So this phosphoglyceromutase is causing the formation of 2-phosphoglycerate from 3-phosphoglycerate. Now what you have seen, from third position, the phosphoryl group has gone to the second position, and this cannot happen without the help of enzyme. So you have the enzyme which is helping in this process. And once this happens, this molecule is quite unstable, and what will happen is that it will release a water molecule. So enolase will come, and a water molecule from this position, one OH from here and one H from here. So this water molecule is released, and what you get is you get enol phosphate, phosphoenol pyruvate. So this molecule is known as a phosphoenol pyruvate. So enol group you have already learned earlier also, that enol group is formed when water is being removed from the structure. So here the phosphoenol pyruvate has been formed, and this phosphoenol pyruvate has much higher phosphoryl group transfer potential, and that is why what it will do is that its phosphoryl group will be transferred to ADP molecule and it will form ATP. So at the end you get a molecule of pyruvate. So it's a phosphoryl group is being transferred, and this phosphoenol pyruvate becomes pyruvate. So this is the end reaction of glycolysis. So glycolysis, you have formed 2 molecules of pyruvate starting from 1 molecule of glucose. Now, just to explain it better, I have already given you all the details how these are happening. The only important point which I would like to emphasize is the enol group that undergoes conversion into a more stable ketone form. So this is a very unstable form. From here to a more stable form is being formed, and this is probably the one which leads to the high phosphoryl transfer potential. And that means there is a large driving force for enol ketone conversion. So you can see the reaction where ATP is formed and the conversion of enol group to the ketone group happens. Okay, to summarize this whole reaction, what you had is that you had 1 molecule of glucose. So this is a glucose molecule. What you got at the end is that, so glucose was 6 carbon. You got 2 molecules of pyruvate. Now, I have written 2pi. If you look at the reaction, you find only one place where pi is involved. There's one ADP which is being consumed, and there is one NADP which is being consumed. But in the reaction, I'm writing twice. Why? Because after pyruvate, these reactions are occurring after formation of glyceraldehyde 3-phosphate, and that's why there are 2 cycles of that reaction occurs, and that's why you have 2pi, 2 ADP, 2 NAD. So once you have 2 NAD plus, they are going to form 2 NADH. When you have 2 ADP molecule, they are going to be phosphorylated, making 2 ATP molecule. And the end of the reaction, what you see is that though there are 2 ATP being consumed, there are 4 ATP being produced. So ultimately, the net result is that you get 2 molecules of ATP at the end of glycolysis. What else you have seen? You have also seen that at the end of glycolysis, you get 2 NADH. Now, once 2 NADH is being produced, now question is that it will create a redox imbalance. Why? Because if I look at these two reactions, of course, because of the proton, there is, they are matching the charges. But otherwise, what will happen on one side, you have the NAD plus, on the other side, you have NADH. Now, this NAD is a limited quantity. Do you remember from where this NAD comes? What is the source of NAD plus? Any one of you? Any idea? Recall we have talked about vitamins, and out of the vitamin there is a vitamin called as nicotine. That is the one which forms NAD. So these are coenzymes. This is what is required with most of the dehydrogenases, and they have to be regenerated. They are not present in abundant quantity. So at the end of the reaction, somehow you have to make it sure that this NAD plus is regenerated. I will discuss this point. If I block the regeneration of NAD plus, the reaction will not happen because this is required. This is required for which reaction required this NAD? This is the reaction. If I don't give NAD, if all of the in the suppose in the cell everything is present in NADH, now this reaction will not happen, and that's why it is important that we recycle NADH. I will talk about there are three different mechanisms by which NADH is being recycled. We call it as a redox balance, and that is what we will talk about. So I have also covered this stage 3, and in stage 3, what you have learned is that two molecules of NADH, two molecules of ATP at this stage, and two molecules of ATP at this stage are being formed. So ultimately, this is the one which is generating process. This is what is being consumed process, and that's the way you say that at the end you get two molecules of ATP and two molecules of NADH. Now this is the complete reaction. You have glucose, ATP is being consumed. This stage again ATP is being consumed. This is the stage where you have required NAD plus. This is the stage where you have produced ATP. This is the stage where you have produced ATP. Okay, so this gives you a basic idea that how glycolysis occurs. Now let us talk about something interesting about maintaining the redox balance. Can you see the slide? Is this
Yes, sir, it's
visible?
visible. It's visible.
Okay, fine. Now, how to maintain the redox balance? The other day, yesterday, I have asked you a question. The question was, why pyruvate is converted into ethanol? Is it dead end product of the metabolism, or there is some metabolic function? That is what we are going to discuss today. So when you have seen the glucose is forming pyruvate, the pyruvate does not exist as such in the cell. Because if you keep on increasing the pyruvate, what will happen? That the acidity will keep on increasing, and that is not good for the health of the cell. So what will happen? That pyruvate, when it is formed, it is converted into different forms. And there are two purposes for this. Number one, that it should regenerate the redox potential. That's very important. Otherwise, the further reaction, glycolysis itself will stop. So there are three reactions of pyruvate which is of importance. One is conversion of pyruvate into ethanol. Number two, conversion of pyruvate into lactic acid. And number three, conversion of pyruvate into carbon dioxide. Now, stage one, the first reaction, conversion of pyruvate to ethanol, and second reaction, conversion of pyruvate to lactic acid. Both of them are anaerobic process, that they do not require molecular oxygen for their reaction. Whereas the last reaction where carbon dioxide is formed, that is the one which is requiring oxygen. So now I throw a question: which process? Suppose you are growing a bacteria, or for that matter, yeast. One is by aerobic process, another is by anaerobic process, in which mass is going to be more, where you will find that more of the mass of the bacteria, or for that matter, yeast, is being produced. Can you tell me the answer? Any one of you? Where do you find more mass? Mass is going to be more in case where you have aerobic process. Why? Because you will get more energy. In case of formation of ethanol, in case of formation of lactic acid, though they can produce quick energy, but that is only 2 ATP. Later on, you will see that there is a large amount of ATP is being produced when they enter and convert the glucose into carbon dioxide. On the other hand, you can think about this in a very different way. When glucose is converted into ethanol, it is a partial conversion. It's a partial oxidation. It's not complete oxidation. Still, you have an organic molecule convert into organic molecule. And that is why we call them as a fermentation process. In fermentation, one organic molecule is converted into another organic molecule, and which is not completely oxidized. So it is incomplete oxidation. And still, your molecule like pyruvate has enough potential to produce energy, and that's where your aerobic process is the one which gives you a lot of energy. Now let's talk about first ethanol fermentation. So you have seen that pyruvate has three fate. Fate number one for ethanol, fate number two for lactic acid, and fate number three is for carbon dioxide. So let's see how these fates are being achieved, how they move on to these reactions. So ethanol is formed from pyruvate in yeast and several other organisms. The first step is decarboxylation of pyruvate. So pyruvate, it's carbon dioxide, this one, that is being removed. And what you get is you get acetaldehyde. The first reaction is happening, and what you are looking at is decarboxylation reaction. So what happens? You have an enzyme, pyruvate decarboxylase, which forms acetaldehyde from pyruvate. Now, this acetaldehyde is converted into ethanol, and when it is converted into ethanol, the NADH, from where you are getting NADH? From glycolysis. So, step where your glyceraldehyde 3-phosphate has been converted into 3-phosphoglycerate, during that step, NADH was produced. That NADH will be reutilized. So, what will happen? It is a process which is for regeneration of NAD+. Now, this NAD+ can again be utilized in that reaction. I'll show you the complete reaction and we'll try to explain it there as well. So, this process of ethanol fermentation is the process of a regeneration of NAD+. Cell does not want to make ethanol. It is not going to become drunk. It is the one which is producing for its own purpose where NAD+ is required, and NAD+ is the one which is important for the continuation of glycolysis. Now let us see. So what happened at the end of the reaction? You have glucose. Whatever NADH was generated has been converted back into NAD+. So that portion is over. So at the end, you have glucose, you have two molecules of phosphorus, two ADP, two protons, and you get two ethanol, two carbon dioxide, two ATP, and two molecules of water. So that's the end of the reaction. Now look at the lactate fermentation. So lactate formation from pyruvate happens in variety of microorganisms. It happens in our body as well. It takes in higher organism when the amount of oxygen is limiting. So suppose you are running, what happens? You are doing a lot of muscle exercise, muscle needs energy, and during this process, it needs quick energy. It doesn't wait for, and second thing is that when you are running, the consumption of oxygen is less so that it does not get enough oxygen to complete the process. And during this process, what happens? The reduction of pyruvate by NADH to form lactate happens. And when you lactate happens, what happens? You get pain. So why do you get pain in muscle? Because of this lactate accumulation. And if you leave it for a certain time, they are again converted back into pyruvate. and once they are converted back into pyruvate, they can utilize the energy. So it's a transient process, and that's why many times you see that the runner, they keep on taking glucose. So the overall reaction of this will be that the pyruvate will be converted into lactate in presence of enzyme lactate dehydrogenase, and this reaction again regenerates NAD+. So this reaction is important for continuation of glycolysis. If glycolysis, if you do not have this reaction, you will not generate NAD+, the glycolysis will not happen. Same type of reaction like you have seen in a case of alcoholic fermentation. Glucose is converted into lactate, ADP is converted into ATP, and 2-PI is converted, is being utilized in the process. The regeneration of ADP you have seen under anaerobic process happening by formation of either ethanol or by formation of lactate. So now let us see what is another fate. Okay, this is the one which is summarizing the reaction. One is where, by formation of acetaldehyde, you have formed ethanol and NAD has been regenerated. Here, pyruvate has converted into lactate, NAD has been regenerated. And third reaction is where what you do is that pyruvate is converted into acetyl-CoA and further oxidation occurs. And here, NAD plus is regenerated, not in this cycle, which is called as a TCA cycle, but it is regenerated by oxidative phosphorylation. We'll talk about that. Now, interestingly, the enzyme involved here, whether you have alcohol dehydrogenase, you have other dehydrogenases, they have excuse me, they are very interesting enzyme. They are the one which are carrying out this reaction, and this is the mechanism which has been given, where the active site contains zinc ion bound to 2 cysteine residue and 1 histidine residue. Zinc ion binds to the aldehyde, and that's the way aldehydic substrate through the oxygen atom, and this polarization occurs, and that's why the hydroid ion is accepted. Now let us move on to further that this dehydrogenase, most of these dehydrogenase, what you have learned is that glyceraldehyde 3-phosphodehydrogenase, which you have seen in glycolysis, alcohol dehydrogenase, which you have seen in fermentation, which is alcoholic fermentation, lactate dehydrogenase, which is involved in the lactic acid fermentation. All of them, they require NAD plus binding. So what you have seen is that the nucleotide binding region is made up of 4 alpha helix. So you have enzyme which can allow the binding, and there are 6 beta parallel strands. Now, common structural domain, one of the first recurring structural domain to recover, and this is known as Roseman fold. The Roseman fold, so this is the one where you have a nicotine binding site and you have adenine binding site. So these two combined is called as a Roseman fold. So Roseman fold is the binding site where your NAD can bind, and it's a common. So you have seen that most of the dehydrogenases you have learned, they have NAD plus binding site or NADH binding site. Now, next question comes that we have talked so much about the glycolysis. What happens to other carbohydrates? So when I eat something, of course, most of the source of energy is starch, and when starch is being hydrolyzed, you get glucose. But we also know that we have a lot of other molecules which we are consuming, maybe even disaccharide like sucrose. So if I break down sucrose, what we get? We get glucose molecule and fructose molecule. Another source of energy could be where we consume lactose. Do you recall where lactose is present? It is a carbohydrate which is present in the milk. So now it's again a question, do we have fructose and galactose being metabolized from the same mechanism, or there are some difference in the metabolic process? You will learn that some way these fructose and galactose, they are interconverted to a stage where they can enter in glycolysis. So first we will talk about how these interconversion occurs, how fructose can enter in the glycolysis. So first thing is that if fructose is there, as I told you yesterday, the fructose can be recognized by hexokinases. That means fructose can be phosphorylated, fructose can form fructose 6 phosphate. And you know that in the glycolysis, first glucose is converted into glucose 6 phosphate, then isomerized to fructose 6 phosphate. So a small amount of fructose can enter in the cycle by formation of fructose 6 phosphate by hexokinases. So it's the same reaction as you have learned in glycolysis. The only thing is that the fructose is entering at different steps. But what happens? The fructose and glucose, they differ in terms of their Km. So Km is as such that if I say the Km of fructose is very high compared to glucose, so what will happen if glucose and fructose, they are present in equimolar amount? What will happen? Glucose will be utilized. Unless complete glucose utilization is not happening, fructose will not be utilized. So fructose will remain unutilized. And that's why what you have is that you have a different pathway by which fructose can be metabolized. So much of the fructose which is being present, they are metabolized by the liver using fructose 1 phosphate pathway. So what it will do is that it will use a different enzyme. It will make fructose 1 phosphate. So first step of phosphorylation is fructose to fructose 1 phosphate, and the enzyme is a similar kind of enzyme, but it has a specificity with the fructose. So it is called as a fructokinase. So in the first step, you will form fructose to fructose 1 phosphate, and this fructose 1 phosphate is split into glyceraldehyde and dihydroxyacetone phosphate. Now you remember glyceraldehyde and dihydroxyacetone phosphate, they are intermediate of glycolysis, and there these products they can enter. So this is again when fructose 1 phosphate is split into glyceraldehyde and dihydroxyacetone phosphate, it is the same kind of splitting mechanism which you have learned in glycolysis, where fructose 1,6 bisphosphate is being split, and the enzyme was aldolase. So here the enzyme is slightly different because the substrate is different. Here substrate is fructose 1 phosphate. There you had a fructose 1,6 bisphosphate. There the product was glyceraldehyde 3 phosphate and dihydroxyacetone phosphate because there was another additional phosphate was present. Now what you have is you have glyceraldehyde and dihydroxyacetone phosphate. Only one phosphate is missing from the product. But again, what is happening is that it is entering in the same glycolytic pathway. Now look at galactose. There is no catabolic pathway to metabolize galactose. So the strategy is galactose should be converted into glucose 6 phosphate. So galactose as such is not going to be consumed. It is converted into glucose 6 phosphate, and there are this complex process. And I would like to give you some hint that how it is happening because it's a very important metabolic reaction. And this also leads to some genetic disorder if some of the enzymes are absent. So let's talk about that. So this complex process, galactose is first converted into galactose 1 phosphate. Like you have seen fructose converting to fructose 1 phosphate, you have also seen that glucose is converted into glucose 6 phosphate. The difference you see here is that whether fructokinase or galactokinase, both of them forming at first position, whereas glucose was forming at sixth position. Now, once galactose 1 phosphate is formed, it is in presence of one molecule which is known as UDP glucose. Now, so far, I have not talked about this molecule is coming for the first time in front of you. So what happens that UDP, can you recall what is UDP? Uridyl? See, you have learned about you have learned about ATP. There you had base molecule which was adenine. Here it is a diphosphate, but the base molecule is uridine. That is the difference. And why it is linked with glucose? Because as you remember that when you have ATP or ADP, they are having high energy molecules. So there is a high energy rich molecule which is formed between UDP and glucose. And when this bond is broken, there is enough energy to catalyze the reaction. Of course, you will require an enzyme. So what you have is that you have UDP glucose, you have UDP glucose, you have galactose 1 phosphate. So there is a transfer which is happening. So what will happen from here? The UDP part will, what it will do is that it will form from galactose 1-phosphate to UDP galactose. So what is happening? Galactose is being transferred here and glucose is being released from here. So this transfer of or exchange of glucose and galactose is happening in presence of transferase, which is known as galactose 1-phosphate uridyl transferase. Don't get scared, because this is then the name of the substrate which you have here, galactose 1-phosphate. This is the uridyl group, uridine phosphate. This is the one which is being transferred, so that is why it is known as uridyl transferase. So what is happening here is one molecule of glucose, one molecule of galactose is present. They will be exchanged, and what you will have is you will have UDP galactose here and glucose 1-phosphate. And this glucose 1-phosphate will further be entering in the metabolic pathway. That part I will tell you later. But here, what you have done is that you have got UDP galactose. Now, do you recall what is the difference between galactose and glucose? The structural difference between galactose and glucose? At the fourth position, galactose is the epimer of glucose. So what will happen? You will see that the position 1, 2, 3, 4. This is the fourth position. Here, this position is changing. So what you have is that you get UDP. From UDP galactose, you get UDP glucose. So that's the way epimerase enzyme works. So actually what happens, the glucose is regenerated. So what you have is UDP galactose can regenerate UDP glucose. So that's what is the reaction which is given here. You have a galactose plus ATP forming glucose 1-phosphate plus ADP plus proton. And this reaction can enter in the pathway. So I will just discuss this point and we will stop here. So you have seen that how fructose is entering in the pathway. The fructose is converted into fructose 1-phosphate. It is then cleaved into two molecules, glyceraldehyde and dihydroxyacetone phosphate. Glyceraldehyde again converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. They will be interconverted, and this will enter in the glycolysis. So here again isomerase will act, and all of them, they will enter in the same pathway. What happens in case of a galactose? If I look at this reaction, galactose will enter to form glucose 6-phosphate. So this is the reaction which will be again entering in the same pathway. So you have seen the glycolysis from different views. Whether we have glucose, galactose, or fructose, all of them, they enter in the same pathway. There may be different mechanism in entering in the pathway. For example, here you have seen glucose, galactose, first converted into galactose 1-phosphate, then you have a transferase enzyme, and then it is epimerase enzyme. So there are a class of enzyme which is helping. I will tell you the importance of this reaction, conversion of galactose to glucose 6-phosphate. If it is not converted, what are the kind of disease you can have? And of course, this process you have already learned that fructose can be converted into fructose 6-phosphate. How does this happen? Just now we have discussed how fructose is converted into fructose 6-phosphate. Of course, there is one reaction where it is converted into 1-phosphate and then cleaved into 2,3 carbon molecule. How fructose directly can be formed from fructose to fructose 6-phosphate? In presence of hexokinase, the same enzyme which was converting glucose to glucose 6-phosphate. Okay, so I think we have discussed the pathway by which glycolysis operates, and we have also discussed how different types of 6-carbon molecule can enter in the pathway, how the redox balance is conducted, how this is fermentation versus your energy generation process is happening. I have not talked about a reaction, this part. This I will elaborate how the pyruvate is converted into acetyl-CoA, because this is a complex enzyme. I need to discuss it more. But you have learned there are 3 processes by which pyruvate can be further metabolized. These 2 processes, ethanol and lactate, is the fermentation process. They are the one which will generate very less amount of energy. And here, this acetyl-CoA, when it is formed, they will be the one which will be for major production of energy. So, and this pathway will proceed only when molecular oxygen is present. Now I'm stopping at this point. Any question? Any one of you? So if there are no more questions, I will sign off. Thank you very much.
Thank you, sir.